Tata Box. In molecular biology, the Tata Box, also called the Goldberg-Cognass box, is a sequence of DNA found in the core promoter region of genes in archaea and eukaryotes. The bacterial homologue of the Tata Box is called the Prevalence box, which has a shorter consensus sequence. The Tata Box is considered a non-coding DNA sequence, also known as a cis-regulatory element. It was termed the Tata Box as it contains a consensus sequence characterized by repeating T and A base pairs. How the term box originated is unclear. In the 1980s, while investigating nucleotide sequences in mouse genome loci, the Hogness box sequence was found and boxed in at the 31 position. When consensus nucleotides and alternative ones were compared, homologous regions were boxed by the researchers. The boxing in of sequences sheds light on the origin of the term box. The Tata box was first identified in 1978 as a component of eukaryotic promoters. Transcription is initiated at the Tata box in Tata containing genes. The Tata box is the binding site of the Tata binding protein (TBP) and other transcription factors in some eukaryotic genes. Gene transcription by RNA polymerase II depends on the regulation of the core promoter by long-range regulatory elements such as enhancers and silencers. Without proper regulation of transcription, eukaryotic organisms would not be able to properly respond to their environment. Based on the sequence and mechanism of Tata box initiation, mutations such as insertions, deletions, and point mutations to this consensus sequence can result in phenotypic changes. These phenotypic changes can then turn into a disease phenotype. Some diseases associated with mutations in the Tata box include gastric cancer, spinocerebellar ataxia, Huntington's disease, blindness, beta thalassemia, immunosuppression, Gilbert syndrome, and HIV-1. The Tata binding protein could also be targeted by viruses as a means of viral transcription. History Discovery The Tata box was the first eukaryotic core promoter motif to be identified in 1978 by American biochemist David Hogness, while he and his graduate student Michael Goldberg were on sabbatical at the University of Basel in Switzerland. They first discovered the Tata sequence while analyzing five DNA promoter sequences in Drosophila mammalian and viral genes. The Tata box was found in protein coding genes transcribed by RNA polymerase II. Evolutionary history. Most research on the Tata box has been conducted on yeast, human, and Drosophila genomes. However, similar elements have been found in archaea and ancient eukaryotes. In archaea species, the promoter contains an 8-bp-AT rich sequence located around 24 bp upstream of the transcription start site. This sequence was originally called Box A, which is now known to be the sequence that interacts with the homologue of the archaeal Tata binding protein. Also, even though some studies have uncovered several similarities, there are others that have detected notable differences between archaeal and eukaryotic TBP. The archaea protein exhibits a greater symmetry in its primary sequence and in the distribution of electrostatic charge, which is important because the higher symmetry lowers the protein's ability to bind the Tata box in a polar manner. Even though the Tata box is present in many eukaryotic promoters, it is not contained in the majority of promoters. One study found less than 30% of 1031 potential promoter regions contain a putative Tata box motif in humans. In Drosophila, less than 40% of 205 core promoters contain a Tata box when there is an absence of the Tata box and TBP is not present. The downstream promoter element DPE, in cooperation with the initiator element INR, bind to the transcription factor TF2D, TF2D, initiating transcription in Tata-less promoters. The DPA has been identified in three Drosophila Tata-less promoters and in the Tata-less human IRF1 promoter. Features: location. Promoter sequences vary between bacteria and eukaryotes. In eukaryotes, the Tata box is located 25 base pairs upstream of the start site that RPB4, RBP7 used to initiate transcription. In metazoans, the Tata box is located 30 pairs upstream of the transcription start site. While in yeast, S. cerevisiae, the Tata box has a variable position which can range from 40 to 100 BP upstream of the start site. The Tata box is also found in 40% of the core promoters of genes that code for the actin cytoskeleton and contractile apparatus in cells. The type of core promoter affects the level of transcription and expression of a gene. The Tata binding protein can be recruited in two ways: by SAGA, a cofactor for RNA polymerase II, or by TF2D, when promoters use the SAGA Tata box complex to recruit RNA polymerase II. They are more highly regulated and display higher expression levels than promoters using the TF2D TBP mode of recruitment. Analogous sequences. In bacteria, promoter regions may contain a Pribno box, which serves an analogous purpose to the eukaryotic Tata box. The Pribno box has a 6 BP region centered around the -10 position and an 8 to 12 BP sequence around the -35 region that are both conserved. A CAAT box, also cat box, is a region of nucleotides with the following consensus sequence: 5GGCCAATCT3. The CAAT box is located about 75 to 80 bases upstream of the transcription initiation site and about 150 bases upstream of the Tata box. It binds transcription factors CAAT, TF, or CTFs and thereby stabilizes the nearby pre-initiation complex for easier binding of RNA polymerases. CAAT boxes are rarely found in genes that express proteins, ubiquitous in all cell types. Structure, sequence, and prevalence. The Tata box is a component of the eukaryotic core promoter and generally contains the consensus sequence 5 Tata AAT3. In yeast, for example, one study found that various Saccharomyces genomes had the consensus sequence 5 Tata ATAATAG3, yet only about 20% of yeast genes even contain the Tata sequence. Similarly, in humans, only 24% of genes have promoter regions containing the Tata box. Genes containing the Tata box tend to be involved in stress responses and certain types of metabolism and are more highly regulated when compared to Tata-less genes. Generally, Tata containing genes are not involved in essential cellular functions such as cell growth, DNA replication, transcription, and translation because of their highly regulated nature. The Tata box is usually located 25 to 35 base pairs upstream of the transcription start site. Genes containing the Tata box usually require additional promoter elements, including an initiator site located just upstream of the transcription start site and a downstream core element DCE. These additional promoter regions work in conjunction with the Tata box to regulate initiation of transcription in eukaryotes. Function, role in transcription initiation. The Tata box is the site of pre-initiation complex formation, which is the first step in transcription initiation in eukaryotes. Formation of the pre-initiation complex begins when the multi-subunit transcription factor 2D TF2D binds to the Tata box at its Theta binding protein, TBP subunit. TBP binds to the minor groove of the Tata box via a region of anti-parallel beta sheets in the protein. Three types of molecular interactions contribute to TBP binding to the Tata box. Four phenylalanine residues, Phe57, Phe74, Phe148, Phe165, on TBP bind to DNA and form kinks in the DNA, forcing the DNA minor groove open. Four hydrogen bonds form between polar side chains on TBP amino acid ASN 27, ASN 117, THR 82, THR 173, and bases in the minor groove. Numerous hydrophobic interactions 15 formed between TBP residues, notably LL152 and LEU163, and DNA bases, including van der Waals forces. Additionally, binding of TBP is facilitated by stabilizing interactions with DNA flanking the Tata box, which consists of GC-rich sequences. These secondary interactions induce bending of the DNA and helical unwinding. The degree of DNA bending is species and sequence dependent. For example, one study used the adenovirus Tata promoter sequence 5CGC Tata AAAG GGC3 as a model binding sequence and found that human TBP binding to the Tata box induced a 97-degree bend toward the major groove, while the yeast TBP protein only induced an 82-degree bend. X-ray crystallography studies of TBP Tata box complexes generally agree that the DNA goes through an 80-degree bend during the process of TBP binding. The conformational changes induced by TBP binding to the Tata box allows for additional transcription factors and RNA polymerase II to bind the promoter region. TF2D first binds to the Tata box, facilitated by TF2A binding to the upstream part of the 2F TF2D complex. The TF2B then binds to the TF2D TF2A DNA complex through interactions both upstream and downstream of the Tata box. RNA polymerase II is then recruited to this multi-protein complex with the help of TF2F. Additional transcription factors then bind, first TF2E and then TF2H. This completes the assembly of the pre-initiation complex for eukaryotic transcription. Generally, the Tata box is found at RNA polymerase II promoter regions, although some in vitro studies have demonstrated that RNA polymerase III can recognize Tata sequences. This cluster of RNA polymerase II and various transcription factors is known as the Basal transcriptional complex. BTC. In this state, it only gives a low level of transcription. Other factors must stimulate the BTC to increase transcription levels. One such example of a BTC stimulating region of DNA is the CAAT box. Additional factors, including the mediator complex, transcriptional regulatory proteins, and nucleosome modifying enzymes, also enhance transcription in vivo. Interactions. In specific cell types or on specific promoters, TBP can be replaced by one of several TBP-related factors. TRF1 in Drosophila, TBPL1, TRF2 in metazoans, TBPL2, TRF3 in vertebrates, some of which interact with the Tata box. Similar to TBP, interaction of Tata boxes with a variety of activators or repressors can influence the transcription of genes in many ways. Enhancers are long-range regulatory elements that increase promoter activity, while silencers repress promoter activity. Mutations. Mutations to the Tata box can range from a deletion or insertion to a point mutation with varying effects based on the gene that has been mutated. The mutations change the binding of the Tata binding protein TBP for transcription initiation. Thus, there is a resulting change in phenotype based on the gene that is not being expressed. Insertions or deletions. One of the first studies of Tata box mutations looked at the sequence of DNA from Agrobacterium tumefaciens for the octopine type cytokinin gene. This specific gene has three Tata boxes. A phenotype change was only observed when all three Tata boxes were deleted. An insertion of extra base pairs between the last Tata box and the transcription start site resulted in a shift in the start site, thus resulting in a phenotypic change. From this original mutation study, a change in transcription can be seen when there is no Tata box to promote transcription. But transcription of a gene will occur when there is an insertion to the sequence. The nature of the resulting phenotype may be affected due to the insertion. Mutations in maize promoters affect the expression of the promoter genes in a plant organ specific manner. A duplication of the Tata box leads to a significant decrease in enzymatic activity in the scutellum and roots, leaving pollen enzymatic levels unaffected. A deletion of the Tata box leads to a small decrease in enzymatic activity in the scutellum and roots, but a large decrease in enzymatic levels in pollen. Point mutations. Point mutations to the Tata box have similar varying phenotypic changes depending on the gene that is being affected. Studies also show that the placement of the mutation in the Tata box sequence hinders the binding of TBP. For example, a mutation from Tata AAA to Kata AAA does completely hinder the binding sufficiently to change transcription. The neighboring sequences can affect if there is a change or not. However, a change can be seen in HeLa cells with a Tata CAA, which leads to a 20-fold decrease in transcription. Some diseases that can be caused due to this insufficiency by specific gene transcription are thalassemia, lung cancer, chronic hemolytic anemia, immunosuppression, hemophilia B laden, and thrombophlebitis and myocardial infarction. Savinkova et al. Has written a simulation to predict the KD value for a selected Tata box sequence and TBP. This can be used to directly predict the phenotypic traits resulting from a selected mutation based on how tightly TBP is binding to the Tata box. Diseases. Mutations in the Tata box region affects the binding of the Tata binding protein for transcription initiation, which may cause carriers to have a disease phenotype. Gastric cancer is correlated with Tata box polymorphism. The Tata box has a binding site for the transcription factor of the PG2 gene. This gene produces PG2 serum, which is used as a biomarker for tumors in gastric cancer. Longer Tata box sequences correlates with higher levels of PG2 serum indicating gastric cancer conditions. Carriers with shorter Tata box sequences may produce lower levels of PG2 serum. Several neurodegenerative disorders are associated Tata box mutations. Two disorders have been highlighted: spinocerebellar ataxia and Huntington's disease. In spinocerebellar ataxia, the disease phenotype is caused by expansion of the polyglutamine repeat in the Tata binding protein TBP. An accumulation of these polyglutamine TBP cells will occur, as shown by protein aggregates in brain sections of patients, resulting in a loss of neuronal cells. Blindness can be caused by excessive cataract formation when the Tata box is targeted by microRNAs to increase the level of oxidative stress genes. MicroRNAs can target the 3 untranslated region and bind to the Tata box to activate the transcription of oxidative stress related genes. SMPs and Tata boxes are associated with beta thalassemia, immunosuppression, and other neurological disorders. SMPs destabilize the TBP Theta complex, which significantly decreases the rate at which Theta binding proteins TBP will bind to the Theta box. This leads to lower levels of transcription, affecting the severity of the disease. Resulting from studies have shown the interaction in vitro so far, but results may be comparable to that in vivo. Gilbert syndrome is correlated with UTG1A1 Tata box polymorphism. This poses a risk for developing jaundice in newborns. MicroRNAs also play a role in replicating viruses such as HIV-1. Novel HIV-1 encoded microRNA have been found to enhance the production of the virus as well as activating HIV-1 latency by targeting the Theta box region. Clinical significance. Technology. Many of the studies so far have been performed in vitro, providing only a prediction of what may happen, not a real time representation of what is happening in the cells. Recent studies in 2016 have been done to demonstrate the Theta binding activity in vivo. Core promoter specific mechanisms for transcription initiation by the canonical TBP TF2D dependent basal transcription machinery has recently been documented in vivo, showing the activation by SRF dependent upstream activating sequences UAS of the human ACTP gene involved in Theta binding. Cancer therapy. Pharmaceutical companies have been designing cancer therapy drugs to target DNA in traditional methods over the years and have proven to be successful. However, the toxicity of these drugs have pushed scientists to explore other processes related to DNA that could be targeted instead. In recent years, a collective effort has been made to find cancer specific molecular targets such as protein DNA complexes, which include the Theta binding motif. Compounds that trap the protein DNA intermediate could result in it being toxic to the cell once they encounter a DNA processing event. Example of drugs that contain such compounds include topotecan, SN38, topoisomerase 1, doxorubicin, and mitoxantrone, topoisomerase 2. Cisplatin is a compound that binds covalently to adjacent guanines in the major groove of DNA, which distorts DNA to allow access of DNA binding proteins in the minor groove. This will stabilize the interactions between the Theta binding protein to the Theta box. This result is to immobilize the Theta binding protein on DNA in order to downregulate transcription initiation. Genetic engineering, Theta box modification. Evolutionary changes have pushed plants to adapt to the changing environmental conditions. In the history of Earth, the development of Earth's aerobic atmosphere resulted in an iron deficiency in plants compared to other members of the same species. Malus baccata var. Xiaojienensis has a Tata box inserted in the promoter upstream of the iron regulated transporter 1 IRT1 promoter. As a result, the promoter activity levels are enhanced, increasing TF2D activity and subsequently transcription initiation, resulting in a more iron efficient phenotype.